Belinda Needham. She received her Ph.D. in sociology from the University of Texas, and then she did a Robert Wood Johnson Fellow at UCSF, and she's here currently as an assistant professor in epidemiology. Her general research focus is on health disparities across multiple lines, including gender, SCS, race, ethnicity, and sexual orientation. She examines social, psychological, behavioral, and physiological mechanisms by which social disadvantage leads to health disparities. And she also works on developing and testing interventions to reduce the effect of social disadvantage on morbidity and mortality. So, welcome.
Thank you. Thank you for inviting me today. I'm really excited to share some of this work with you, and I'm glad that people are interested in telomeres. And so I hope that we will have a really interesting discussion at the end of this. So what I want to do today is start out by telling you a little bit about my story, not just how I started studying telomeres, but how I got interested in sociology, and then how I got interested in biology, and then how those things sort of came together. And then I want to talk to you a little bit about telomeres, what they are, and why I study them, why I think they're interesting. And then I want to tell you, this will be sort of the briefest part of the talk, some study results. I want to show you the results of 2 recent papers that I did on socioeconomic status and telomere length, one study in children, one in adults. And then I want to spend some time talking to you about new projects that I'm doing. So I've got 4 new telomere projects that are all at various stages. So it would be really interesting to hear what you guys think about those projects and to get your feedback and hear your ideas. And then I want to leave a lot of time for discussion and questions. Okay, so I think it's really hard to separate our personal biography from our intellectual biography, and so I'm going to mix them up here and tell you a little bit about me as a person and me as a scholar. So I was born in Birmingham, Alabama in the 1970s. I can see you doing the math. How old is she? So who in this room knows what Birmingham, Alabama in the 1970s and before was known for? Anyone? What's that? Sorry, I can't hear. Does anybody know? This is a hint.
Steel.
Steel. Yeah, steel was the biggest industry in Birmingham for a long time. So my dad was a steel worker. This is my dad here, circled. I don't know if you can see him, but that was my dad. My mom was a stay-at-home mother. And some other stuff started happening in the 1970s in the US. So who among the social scientists here can tell me what was happening in terms of like macroeconomic things around this time?
Inflation.
Inflation, yes. Outsourcing. Yeah, I think my parents told me their first mortgage, they had like a 13% interest rate or something like that. Maybe it was even higher, I don't remember. But yeah, there was inflation. And there was a shift from a manufacturing economy to a service economy. So there was a lockout at US Steel in the early 1980s. I think it was 1980, 1981. And so at that time, the workers couldn't go in. They couldn't work. It was a little different than a strike. And so my parents had to figure out something else to do. So during the daytime, my dad worked at a paint store, and at night he worked as a security guard. And my mom put my then 6-month-old baby sister in daycare, and she went and got a job as a secretary. And between the 3 of them, they could not even come close to what my dad had been making as a steel worker. So this was a really sort of big thing in my life and in the lives of many, many people who lived in this place at this time. So I understood very early that social forces can have a powerful impact on the lives of individuals. And I think that probably sparked my interest in sociology well before I knew what it was. So my parents were very acutely aware that their opportunities were limited by lack of education. Neither of them had gone to college, so then when the shift happened in the economy, it was very hard for them to transition and be part of this changing economy. So they were quite determined that my sister and I wouldn't face that same limitation, perhaps a little over-determined. They were kind of annoying about it. And so when it was time for me to go to college, can anybody guess what my parents wanted me to do?
Engineering.
Oh, engineer, that's a good one. Did anybody say I think I heard doctor in there, right? Oh yeah, medical school. That was the way to have a secure future and a good life, was to go to medical school. But I was really not interested in chemistry or biology or any of that stuff. I was interested in social justice. I was kind of a cliche. So I wanted to go to work for the ACLU. That was my dream. I was going to be a lawyer for the ACLU. So I actually decided to major in sociology because there was a 1-paragraph blurb about it that I got from Texas A&M University, which is where I was going to school, that said it was good preparation for law school. I said, all right, I'll do that. So probably not the best way to choose a major. But we didn't have the internet, so that was what I did. So I took an intro sociology course my first semester in college, and I absolutely loved it right from the first day. I knew this was what I wanted to do. And I have to say that a lot of the credit for this goes to my intro sociology professor, Dr. Cohn. That's him up there in the corner. He looks like a sociology professor, right? Dr. Cohn was awesome. He was very passionate about sociology and really, truly committed to undergraduate education, which we all know is not always the way intro professors are. So I was very fortunate to have him. And in this class, he had us read Suicide. Not an excerpt, not a summary in a textbook, Suicide. He had, first-year college students read this. And I was absolutely blown away by the idea that something as intensely personal as suicide could be socially patterned. And so this was just amazing to me. All right, so then I graduated and I had no idea what I wanted to do. I didn't want to be a lawyer anymore because being a lawyer meant going to law school, and I didn't actually want to do that. So I wasn't sure what I wanted to do. I took a year and decided to work. And so I got a job working at the Texas Department of Human Services, where I determined eligibility for TANF, food stamps, and Medicaid. And I was thinking at the time I wanted to go into public policy, and this would be a great experience. I could learn something about policy. Well, while I was there, I really had this overwhelming sense that I was doing more harm than good, and I didn't want to do this anymore. And I realized that even though when I graduated from college, I said, the one thing I know I will not do is get a Ph.D. in sociology. That's the one thing I will not do. I'm not sure what I will do, but that's the thing I'm not going to do. I realized after about a month of doing this, that was what I had to do. I had to go back, I had to get a PhD because I had to understand what was causing social stratification, where was this coming from, and what could we do about it. So then I went to grad school. And after I took a sociology of health and illness course, I ended up getting really interested in the consequences of social stratification. I'd never really thought about that before. I always thought of social stratification as the outcome, not as the exposure. And I was, again, I'm easily fascinated. I was fascinated by the fact that your social status could have an effect on your physical health. This had never occurred to me. So I want to take just a few minutes to show you some of the evidence for what I'm talking about. And you guys already know this, but I think it's good to see it again, just to remind us of how profound these associations are. So in the next few slides, I'm going to show you some data on educational gradients in morbidity and mortality in the US. And I'm going to start with data for adults and then show you a little bit of data for kids. We don't have much, but show you what we have. So this is self-rated health. This is data from the National Health Interview Survey. So this is for self-rated health, and you can see the percent of respondents that are reporting excellent or very good health are the blue bars. And you can see that as the level of education increases, the percent of people reporting excellent or very good health increases. And you can see the opposite pattern for reporting fair or poor health. As education increases, fewer people report fair or poor health. All right, there's similar patterns for age-related chronic diseases. I'm going to go through several and show you. This is coronary heart disease. This is self-report. Has a doctor ever told you you have coronary heart disease? This is respiratory disease, so you see the same pattern for emphysema that you saw for coronary heart disease. This is the data for stroke. These really are different figures, too. These aren't the same one. Diabetes. And then this one's a little messier, so this is mortality. And although the educational gradient isn't quite as clear for mortality, you can see that the age-adjusted mortality rate is much lower for those with at least some college than it is for those with a high school diploma or less than high school. All right, so now I want to show you just a little bit about kids. We don't have as many ways to assess health status in kids, but you can see similar patterns. Here, this is for parental education and the parent report of the child's health. So the percent of parents reporting that their child is in excellent health, that's the blue bar. It increases as the level of education increases. And the percent reporting that their child's health is fair or poor, the purple bar, that decreases as education increases. And here's data for infant mortality. So fortunately, infant mortality is a rare event, but you can see here a clear educational gradient with the highest death rate for infants born to mothers with less than a high school education. All right, so just to summarize, increased educational attainment is associated with decreased morbidity and mortality. Education is considered a fundamental cause of disease because it's associated with multiple disease outcomes, as I just showed you. Also, it operates through multiple risk factors, including material disadvantage, stress exposure and vulnerability, and health behavior. And it involves access to resources that can be used to avoid risks or minimize consequences of disease once it occurs. All right, so all of this was really great and really exciting, but I felt like something was missing. And that missing piece was biology. If social circumstances influence our physical health, they have to do it somehow. They have to get into our bodies somehow to influence our physical health. And at this point in my training, as I was finishing up graduate school, I hadn't learned anything about the biology. And so that was really the missing piece for me. And while I was working on my dissertation, I started reading all of this really exciting theoretical work on biological mechanisms. So we had Arlene Geronimus's work on weathering, Bruce McEwen and Teresa Siemens' work on allostatic load, Clyde Hertzman's work on biological embedding, and Nancy Krieger's work on embodiment. And I really wanted to incorporate these ideas into the work that I was doing, and I didn't know how. So I applied for an interdisciplinary postdoc in population health to learn more about the biological mechanisms that link social status to physical health. And I was very fortunate. I got to spend 2 years at UCSF and UC Berkeley in the Health and Society Scholars Program, where this is their little picture that they put all over everything, where the emphasis was on understanding the links between social context, behavior, and biology from a life course perspective. So this fit perfectly with all the things that I had been doing, just added the biology piece in there. And this is where I met Alyssa Eppel, who's here, and Nancy Adler, and Zhu Lin, and Liz Blackburn. These people were part of my mentoring team while I was a postdoc, and these were the people that first exposed me to work on telomeres. All right, so what are they? Telomeres are the protective caps at the ends of chromosomes. They get shorter every time a cell replicates. They also shorten with exposure to oxidative stress. Now, telomerase can counteract shortening by elongating and protecting the telomeres, but it's expressed at very low levels in most of our cells, and that is in part to protect us from cancer. Once the telomeres become too short, cells become senescent and they will no longer divide. Older people tend to have shorter telomeres than younger people, although there's a lot of inter-individual variation in telomere length, about half of which is thought to be genetic. In most, but not all, epidemiologic studies, short telomere length is associated with age-related diseases, including cardiovascular disease, type 2 diabetes, dementia, and cancer, and also with mortality. And these are independent of chronological age. So telomere length is hypothesized to be a biomarker of organismal aging. Although, of course, there's some debate about this and some skepticism about it, and that is something I'd be happy to talk about with people later if you're interested in that. Okay, so now I want to tell you why I got interested in telomeres and why I still study telomeres and think that they're interesting. So I think they have some big advantages over other biomarkers and other health-related outcomes that we might study. And so some of these are practical reasons why telomere length is a nice thing to study. And one is that it can be measured in easily accessible tissues. So we can measure this in blood or saliva. And it can even be measured in stored samples as long as they've been stored under certain conditions. And so that's really nice for the type of work that I do. Also, telomere length is a single measure. So it's easier to get the data for this and easier to analyze than allostatic load, for example. I don't know if you guys have worked with allostatic load before, but it's usually made up of a number of markers. It can range anywhere from 4 or 5 markers to 10 or 15 markers. And you have to add all these things up, and they're measured on different scales, so you usually have to dichotomize them and then add them, and it's challenging to get these data. Telomere length is also stable in the short term, so it doesn't depend on the time of day that you collect the sample, unlike some things like salivary cortisol that do depend on the time of day and can be really tricky to analyze. It's relatively inexpensive, so right now for the studies that I'm doing, I'm paying about $25 per sample for the assay. That's as long as the DNA has already been extracted, so that's pretty good. And so those are the practical reasons. But then there's also some theoretical reasons why I think telomeres are really nice to study. The one that's most important to me is that telomere length is not a risk factor for any specific disease. It increases the risk of many disease outcomes. And this is important, especially if you're studying socioeconomic status, because we know that the effect of low SES on health is nonspecific. As I showed you earlier, it's related to all kinds of outcomes. We know that telomeres change in response to environmental exposures. If this were entirely genetically determined, it wouldn't really be that interesting to study in terms of social exposures. It's also preclinical. So it can provide evidence of disease risk prior to the onset of clinically significant disease. So this makes it possible to study telomere length in healthy populations, including young populations of children, adolescents, and young adults. Okay, so hopefully I've convinced you that telomeres are interesting and important. And now I want to show you the results of a couple of previous studies that I've done on telomere length. All right, so in the first study, we looked at the association between parental SES and child telomere length. This was one of the first published studies on telomeres in children. There's now been several. We've known for quite a while that childhood SES is associated with age-related disease and mortality in adulthood. But the extent to which socioeconomic disparities are already evident during childhood is somewhat less clear. That's because it's hard to assess disparities during childhood. Kids are generally pretty healthy, and the kinds of problems that they tend to have, things like asthma, injuries, dental caries, aren't really the kinds of problems that we tend to care about in adults. So in adults, we're interested in diseases of aging primarily because these are the leading causes of death. So we're interested in things like CVD and diabetes, which are not common among children. Because telomere length is associated with age-related disease and mortality in adults, the idea for this study was that if we could observe differences in telomere length among kids, this might help explain why we see associations between childhood SES and morbidity and mortality later in life. So the data for this study were from the Admixture Mapping for Ethnic and Racial Insulin Complex Outcomes Study, or the AmeriCO study. This was a population-based study of healthy children in Birmingham, Alabama. The data were collected between 2005 and 2008. And for this study, we randomly selected 35 white and 35 African American children from the full sample of 360 kids who were aged 7 to 13. We used the PCR method to measure relative telomere length in stored DNA samples. Okay. So compared to children with at least one parent who graduated from college, kids whose most highly educated parent never attended college had significantly shorter telomeres. The sample had relatively high educational attainment among the parents. So 70% of the sample had at least one parent who graduated from college. And so those whose most highly educated parent never attended college was only about 13% of the sample. And so then we looked at some potential mediators of this association. And we found that it was not explained by diet. We had measures of fruit and vegetable consumption, physical activity, or body mass index. So the next study I want to show you is one where we examine the association between adult SES and telomere length in the haines study. So prior work in this area, the area of adult SES and telomere length, had produced mixed results. And most of the studies found an association between at least one indicator of SES and telomere length, while several other studies found no association at all. And it had been suggested at the time that conflicting findings may have been due to the use of small non-representative samples. So this was the first study of telomere length in a large, nationally representative sample. And we started the work for this project during my postdoc, is when we applied for the grant to do this work, and then it was several years later that we eventually got the funding and were able to do this study. But the data are now publicly available, so they're actually on the haines website. If anyone is interested in using those data, I'd be happy to talk to you about them, share code, whatever. So it's a nice resource, although it can be a little overwhelming to just get that data set without any background information. But we do have, we have put together some documentation for the website that describes the method that was used and all that. So hopefully it's pretty user-friendly. So these data are from haines 1999 to 2002. We used stored DNA from purified whole blood for almost 8,000 adults aged 20 to 85 plus. Again, we used the PCR method to measure relative telomere length. And the analytic sample for this study is just over 5,000 people. It includes white, African American, and Mexican American participants aged 20 to 84. So we found that compared to those with a college degree, those who completed less than high school had shorter telomeres. And this association was not significantly different across race ethnic groups. And again, here we decided to look at some potential mediators, and we found that the association was partially mediated by smoking and body mass index. Okay, so here's just a list of some other telomere papers that I've done in the last couple of years, and I'm happy to talk about these with you guys if you're interested in some of the results. But so we have a paper where we looked at telomere length and mortality in haines. So haines is connected to the National Death Index, and we were able to look at mortality. We had, I think, about 850 deaths that occurred over almost 10 years of follow-up. And so this paper is in press right now. I actually was, before I came over here, I was doing edits to that one, so I remember the results in case anyone wants to ask. These are not overwhelmingly exciting results. We found sort of limited support for the idea that telomere length increased the risk of all-cause mortality. We found stronger findings for cardiovascular mortality, which is something that has been sort of coming out in the literature over the past several years, is that it seems like telomere length is a really nice biomarker for cardiovascular health, perhaps more so than for some other outcomes. We did interestingly find that there were interactions with race, particularly for cardiovascular mortality, such that telomere length was a stronger, better predictor of mortality, cardiovascular mortality in African American respondents than in either white or Hispanic respondents. I'm not exactly sure what this means, but for those of you who are doing work in this area, you know that it is a really common finding, and we've found it in haines, and I believe it's been found in HRS also, that African Americans have longer telomeres than whites. And so people have been trying to understand this. Why is this the case? Since African Americans have higher morbidity and mortality than whites, why would they have longer telomeres? And so I think this may help us understand it a little bit, although I'm not exactly sure. We also have a paper on depression, anxiety, and telomere length that came from the haines data. So haines administers the CD to a half sample of respondents aged 18 to 39. So it's a pretty small sample. I believe we had about 1,700 people. We didn't find any direct effects of depression or anxiety on telomere length, but we did find a significant interaction with the use of antidepressants. It was kind of an interesting finding, and we think that what it suggests is that people who still meet the criteria for major depression, despite being treated for depression, had significantly shorter telomeres. So this might be depression that's resistant to treatment.
I'm sorry, I didn't understand what you just said.
Okay.
What's the relationship to antidepressants?
So with telomere length,
Yeah.
there's no association. So people who take antidepressants do not have, like, there's no direct effect of antidepressant use on telomere length.
Okay.
It's an interaction with currently meeting the criteria for major depression.
Whether or not they're taking antidepressants.
Sorry, say that again.
The correlation is with currently meeting criteria, whether or not they're taking antidepressants.
So it's an interaction. So there's an interaction between current antidepressant use and meeting the criteria for major depressive disorder in the past 12 months.
So you're suggesting that's a particularly resistant form of depression or anxiety because they still meet criteria while they're on medications. That's associated.
And that's associated with shorter telomeres.
Thank you.
Sure. Does that make sense? It's a guess. We don't know, but we're trying to understand what that interaction means. I was also part of a paper with an environmental epidemiologist that looked at lead and cadmium exposure and telomere length. So haines has this sort of interesting data on environmental exposures. And they found that not lead, but cadmium exposure was associated with shorter telomeres. And that was largely explained through smoking behavior. So apparently cadmium is involved in smoking. I don't know much about environmental things. We also have this paper on sugar sweetened beverages and telomere length that was led by a nutritional epidemiologist. This paper got a lot of press and a lot of criticism, and was an interesting experience. And then I've also been doing some work in the past couple of years since I came to Michigan on telomere length in the MESA study. That's the multi-ethnic study of atherosclerosis. And so I have a paper that came out last year in biodemography and social biology where we looked at gender differences in telomere length, and I was particularly interested in looking at mediators of that association. So there's been a lot of speculation that so women have longer telomeres than men, and a lot of the proposed hypotheses for this are related to biological factors, particularly sex hormones, which you're probably not surprised by. Anything that's gender differences is usually attributed to sex hormones. And so we actually had data on sex hormones in MESA because it's a cardiovascular study and they were interested in the hypothesis about estrogen being related to cardiovascular disease. And so we looked at gender differences and gender by age differences. And so the gender by age term suggested that women experienced a faster cross-sectional rate of shortening. So this was not longitudinal data. So we'll get to that in a minute about the longitudinal data. But so I was interested in trying to explain the gender difference and also the gender by age difference that we observed, that interaction. And it looked like the sex hormones didn't really explain anything. Which could be because these women were postmenopausal and had very low levels of sex hormones, and so maybe that's why it wasn't explaining anything. But it did appear that the behavioral factors were explaining a lot more of this difference. So in particular, smoking, gender differences in smoking were explaining a lot of the difference in telomere length. And then we also did a paper on neighborhood disadvantage and telomere length that came out last year. And this was a very simple sort of measure of neighborhood disadvantage, just looking at things from the items from census, so sort of like neighborhood socioeconomic status. So the point of this, sorry, I forgot to mention that, was that these SES papers that I showed you are just part of a larger body of work on the social antecedents and health-related consequences of short telomere length. So that's kind of how all these things fit together. Because I feel like at this point we still don't know a lot about telomeres and what the correlates are, and so I think that we need to have this sort of phase of descriptive work looking at these things, and so that's what my work in telomeres has focused on sort of up until now. But then I want to tell you about the new projects, and this is sort of like the second phase of thinking about telomeres. So the first study that I want to tell you about is part of the blood spot environmental epidemiology program here at the University of Michigan. Coulter Mitchell, who's in the back, is a co-investigator on this project. And for this project, we're using newborn dried blood spots from the Michigan Neonatal Biobank. So when a baby is born, they take blood to test for genetic defects, and they store that blood. And then researchers can request access to those blood spots to do studies. And so we requested access to these blood spots to examine how maternal socioeconomic status, that we get that data from the birth certificates, we have maternal education, and we have the type of payment for delivery, so we know whether the delivery was paid for by insurance, by Medicaid, or self-pay. We're using that as a proxy of socioeconomic status. And so the idea for this study is to relate those maternal characteristics to newborn telomere length, to see if we can see differences already at the time of birth. We know we can see them by the time kids are 7 to 13, but can we see them at birth? And this study was motivated by two sort of recent advances. One recent study, very small study, that showed that prenatal exposure to stress, the mother was stressed while the baby was in the womb, and that influenced newborn telomere length. And then also sort of an exciting development, a methodological paper that came out that showed that you could measure telomere length in DNA extracted from dried blood spots. So this study is happening right now. It's primarily a feasibility study, and then we're going to be thinking about ways to extend this to a larger group of people. Another study that I have going on right now is with the element cohort. This is the Early Life Exposure in Mexico to Environmental Toxicants project. I love how people can come up with these. I can't do this, it's a special talent. But so this is a project that's really interested in how exposure to environmental contaminants influences pubertal development and obesity progression during pubertal development. But the reason I got interested in this study is because they have data on sex hormones because they're interested in puberty. And I had just finished my MESA paper on gender differences in telomere length and being kind of frustrated by the fact that, like, oh, well, you know, this isn't really the right age group to be looking at this hormone hypothesis in because these women are postmenopausal. And so this is super exciting because these are girls who are going through the pubertal transition. This is when those gender differences in telomere length are hypothesized to emerge. And so I really wanted to be able to get data on telomere length from the stored blood samples that they have. I just got that data. They're also going to be following up this cohort 2 more times, so there's the possibility of getting longitudinal data. So if we see sort of interesting things with the cross-sectional results with sex hormones and telomere length, then I'm going to apply for funding to go back and do this in the following 2 waves. All right, so the third study, so that group is aged 8 to 15 at baseline. So the third study I'm working on right now is with the National Longitudinal Study of Adolescent Health, or AdHealth. I just submitted an ancillary study application to add telomere length during wave 5. So they're about to go into the field for wave 5. They have stored DNA from wave 4. And so we would be proposing to use the stored DNA from wave 4 plus the newly obtained DNA at wave 5 to look at longitudinal changes in telomere length during young adulthood. So, if the study gets approved, we'll be looking at psychosocial stress exposure as a predictor of change in telomere length over about an 8-year period in young adulthood. And then the last study I wanted to mention is another study with the multi-ethnic study of atherosclerosis, where we went back and we got longitudinal data. So the original studies that I showed you here were just from baseline telomere length. We now have data at baseline and exam 5, which was 10 years later. These people were 45 to 84 at baseline, so this is an older group. But we're going to be able to answer a lot of really interesting questions about the rate of change over time and predictors and sort of consequences of that rate of change. So I want to talk a little bit about issues in telomere research, challenges in telomere research, and maybe future directions, things that I think we should be doing in addition to the longitudinal piece, which is some of the stuff that I showed you earlier. So first, I think we need to be doing longitudinal studies. Almost everything that we know about telomeres right now comes from cross-sectional research. So one of the first things that I want to do with this MESA data that we got a week and a half ago is to look at how the cross-sectional rate of shortening compares to within-person change in telomere length over time. I think that would be really interesting and really important. We know those two things aren't always the same. How much variability is there in the rate of change? What accelerates shortening? What protects against it? Does the rate of shortening predict outcomes better than telomere length at one point in time, or is it just as good to have cross-sectional data? Are there sensitive periods? Do the effects of risk or protective factors depend on life stage? I think being able to combine the data that we have from Ad Health with data from Element, that's the younger cohort, with data from MESA will be really interesting to look at these, really across the life course. How do we account for regression to the mean? This is going to be a big issue in these studies. So the few studies that do exist right now looking at longitudinal changes in telomere length have found that the rate of change greatly depends on the baseline telomere length. And so what is the appropriate way to adjust for that and to model that? That's going to be sort of a tricky issue and one that we're going to be working on. Tissue specificity and cell type. Does it matter what type of tissue we use? Does it matter if we use whole blood versus saliva versus whatever else, from an actual organ or from the skin or whatever? There have been some small studies that have shown that telomere length does vary across tissue type, but it is highly correlated across tissues. So this is sort of going to be an issue in the future. Does it matter what type of tissue we use? Also, a really important thing is the cell type question. So subpopulations of cell types have different average telomere length and different rates of shortening. So within a person, the composition of cell types in white blood cells, which is what we're normally measuring telomere length in, is white blood cells, leukocytes, depends on things like stress exposure and exposure to infectious agents. So the things that we're interested in exposures are influencing the distribution of these cell subtypes, and so that can be potentially problematic. So then the question is, should we look at telomere length just in monocytes or just in some other sort of cell subpopulation? And if we do that, it means that steps have to be taken at the time of data collection to isolate those cell subpopulations. It's more expensive, it's more difficult, it's more time consuming, but is that what we need to be doing? In the field of DNA methylation, which I've also been doing some work in, this is where that's going, that studies are taking place where they're just focusing on monocytes, for example. Measurement. I think this is the big question right now, is measurement. So the PCR technique that I mentioned earlier, which is what most of the big epidemiologic studies have used, they use it because it requires a small amount of DNA and it's relatively cheap. But it has lower reproducibility than some other methods like Southern blots. And this may contribute to what some people have observed in smaller longitudinal studies with a relatively small period of time between sample collection. Oftentimes in those studies, you'll see that about 30% of people have telomere lengthening. And that is sort of contrary to the telomere hypothesis of aging, right? We shouldn't have a third of people experiencing lengthening. And so Abraham Aviv in particular has done some work on this issue and trying to understand it. And he has shown that most of what you observe for lengthening can be related to measurement error. And so he suggested that one thing that we need, along with better measures, is that we need to have a longer period of time between samples. So looking at telomere change over 3 years is probably not going to yield very useful results. You probably need to look at more like 10 years or longer. Another big question, should we be looking at average telomere length, which is what PCR does, or should we be looking at shortest telomere? So which is biologically relevant? It's not really known whether senescence is triggered in the presence of shorter average telomere length or in the presence of one critically shortened telomere. So we may be really measuring the wrong thing altogether. And then telomerase. Mentioned that at the beginning. So telomerase is this enzyme that can lengthen and protect telomeres. So at first people thought that this lengthening that we were seeing in these longitudinal studies, this was real. This was telomere length, or this was telomerase that was doing this. And in theory it can do this, right? But maybe not because of the measurement error. But very few studies have measures of both telomere length and telomerase. And prior research has shown that the combination of short telomeres and high telomerase activity may be indicative of greater cell stress than short telomeres alone. So maybe we need to be thinking about telomeres and telomerase as a system and looking at them in relation to one another, and that will give us more information about health and health-related outcomes. So I just want to acknowledge a lot of people. These are collaborators on the studies that I talked to you about today. So as you would expect, it requires a lot of people to do these kinds of studies because you need a lot of people with a lot of different kinds of expertise. And so this is one of the challenging but also exciting things about working in this area. And then I want to take questions, but I want to show you one thing first before I take questions, and we can talk about this. And I should have just put it in my slides. Has anybody seen this before? The hype cycle. It's about new technologies, but I think it sort of applies to this work on biomarkers and this biosocial research that we're doing. And the idea is that you have some new technology or something, a new biomarker, let's put telomere length here. It comes out, people get super excited about it, right? Because they think, oh my gosh, this is the perfect biomarker. This is going to explain everything. We won't have to do any more tests at the doctor's office. We'll just get telomere length. We'll know exactly what's going on with people. And it leads to the peak of inflated expectations, right? And then what happens is that studies come out and you get results that conflict, right? You get contradictory results in these studies. Things aren't making sense. It's not perfect. It's not what we expected. And this then leads to us entering the trough of disillusionment, where we now hate telomere length and we think it's really stupid because it wasn't as awesome as we hoped it would be. And so I think we've hopefully reached this. I think that we are coming back up to what is, I like the slope of enlightenment, and realizing that maybe telomere length is useful. It's not as perfect as we thought it was going to be, but it is useful, and we need to keep working on it because we don't actually know enough yet to say this is stupid, and we need to move on to something else. That this is a brand new thing, and we really need a lot more work to know if it's going to be useful. And so hopefully we're getting to this plateau of productivity, and telomere length will turn out to be something really useful and important for us to study. Okay, I'm going to stop because I want to take questions. Okay, let me go here. I'm supposed to come near you so that you can hear
So
them.
none of the studies you were presenting were having telomere length as the outcome.
Right. Mm-hmm.
What would a reasonable hypothesis be in terms of using telomere length to predict another outcome? Is there something that we should be focusing on that makes the most sense? I mean,
Yeah.
disease perhaps makes sense.
Yeah.
There are other things like psychosocial outcomes of individuals that might make sense.
Yeah, so the question is about how most of the work that I presented here is looking at telomere length as the outcome, but should we be looking at telomere length as an exposure? And if we do, what would the outcome be? I was focusing more on exposures because there had already been sort of a body of work that was looking at outcomes, specifically looking at these disease outcomes like you mentioned. So there's been quite a bit of work looking at cardiovascular disease, a little less on diabetes, although there's been some. There's been work on dementia and Alzheimer's. Trying to think of what else has been done. Cancer, there's been some work on cancer. Now these are generally smaller studies, but there had been kind of a convincing body of evidence there, and also on mortality. I think the best outcome you can look at if you want to provide evidence for this being important and something we should care about is mortality. And so we did look at mortality as an outcome in the haines study. And we found some suggestion that telomere length was associated with increased risk for mortality, but it wasn't that strong of an association. We only had about 9 years of follow-up. So that may have been part of it. But I think that would be really important to look at. And in the ad health study, I'm trying to think about this. What would be a useful thing to look at in a young adult population? Or children, right? What would you look at? And so I'm really sort of starting to read more and learn more about telomere length and diabetes. And because diabetes is something that can affect a younger population, and you can also measure, there's some preclinical measures, so you can look at HbA1c. And so one of the things that I'm thinking about incorporating in that proposal is to look at telomere shortening and change in HbA1c. So I think that would be useful. Did you have other ideas? Yeah.
Sleep studies.
Yeah.
And newborns are at the beginning.
Yeah.
So if you find that socioeconomic status is indeed related to telomere length of newborns, where does it take you?
Yeah. Right.
Understanding that child's development. I guess that's my point.
Yeah, I mean, we would really need to follow those people over time to understand what happens. I mean, we can sort of extrapolate from what we know about telomere length in mid to late life and disease outcomes and say, okay, we know that telomere length in midlife is related to increased risk of developing diabetes. Therefore, we can assume that if children have shorter telomeres at birth, they're going to have shorter telomeres later in life and will be at greater risk of diabetes. But that's maybe not true. And so ideally, we would really want to be able to follow kids from birth into mid to late life to see what are the disease outcomes that are happening. It would be fantastic if we had a cohort that had stored DNA from birth, but then they're older now and we can start to measure some health outcomes. I mean, it's possible that with the blood spots, we could do some of that. I think they go back to, I want to say they've been stored since the early 1980s, so that population isn't very old yet, but it's possible that within 20 years you could see some, if you could find them and link their data there. So you had a question up here.
Yeah, what did you find with the sugar drink study?
So in that in that study, they looked at sugar-sweetened beverages in different categories. So there were carbonated sugar-sweetened beverages and non-carbonated, so things like Gatorade versus things like Coke. And then also they looked at fake sugar beverages, so Diet Coke or whatever, and it was the sugar-sweetened soda consumption that was associated with shorter telomere length, but the fruit juice and the non-carbonated, like the sports drinks, that kind of sugar-sweetened beverage was not. Right, which is weird, because if it's the sugar, then it should be happening whether it's carbonated or not, right?
Did you control for quantity?
Yes, so that's part of it. So that's one of the things that she speculated, was that there's very differences in the amounts of consumption on average that people have of these drinks. So it was pretty rare at the time of this study, 99 to 02. People didn't drink as many uncarbonated sugar-sweetened beverages as they do today. And so she thought, well, maybe that's part of it. I think it's residual confounding. I think it's something that we couldn't adequately control for, that's associated with both soda consumption and short telomere length. But we controlled for everything we could, and it didn't go away. We controlled for poverty-income ratio. We had tried control for education. Didn't explain it. Yeah.
How good is the evidence that telomeres can't grow? Because it's very depressing to think that in little kids,
yeah,
they're already shorter.
Yeah.
But it sort of makes me suspicious that 30% of people are written off as measurement error, because that's not what we believed to begin with.
Yeah, so the evidence on telomere shortening, or telomere lengthening, I think there is some laboratory evidence that it can happen in animals, that you can lengthen telomeres. And we know that telomeres lengthen in cancer. And that's one of the problems with cancer, is that the telomeres lengthen, and so then the cells replicate and divide in an uncontrolled way. And that's actually bad. So we want to keep telomerase in check. We don't want to just give people drugs to increase telomerase, because then we'd all have cancer. So that's not healthy. I do think that there is the possibility of some real lengthening. And there have been, actually, now that I think about it, there were a couple of studies that were intervention studies, very small studies that were done in California with meditation and diet changes, and they looked at telomere lengthening over a relatively short period of time, so like 6 months. Now, that could be measurement error, I don't know, but it was a randomized study, and they saw that there was greater lengthening in the people that had the intervention. So I think it's possible. I think it's unlikely that 30% of people would have lengthening. That seems unlikely, but maybe. Yeah.
I have a question. I kind of feel like the telomere is kind of like a biologic middleman between like an exposure and then oxidative stress and then telomere shortening and then at the other side of that, senescence. So is telomere shortening better than either an oxidative stress marker or a senescence marker at indicating either of those things?
So what are you thinking in terms of a senescence marker?
I was thinking of some really cell-specific kind of things that are difficult to measure on like a bigger scale. So you think cost-effective-wise it's just?
Yeah, I think that would be hard to do in these types of population studies. And that is one of the debates that I alluded to, and I think I sent around a paper that was talking about that. Like, is telomere length just a biomarker? Is it mechanistically involved in aging? And that's one of the kind of big debates. For my purposes, I don't really care, because of what I'm interested in. But for the biologist, that's really important. Like, is this mechanistically involved? And I think there is some evidence from laboratory studies that it is mechanistically involved in shortening. I will say that I've looked at a marker of oxidative stress in relation to telomere length in the MESA study. So we had GGT, gamma glutal something, and it was not associated with telomere length, which I think was a little strange in that sample. So I don't know how good those markers of oxidative stress are, but I don't necessarily think that telomere length is better than a marker of oxidative stress or senescence. I think it's just something that we have ways to measure. Yeah.
Do you know
how telomere length is controlled in terms of genetics or epigenetics or anything like that?
Yeah, so there's been some studies that have looked at genetic factors, and so there was a GWAS that came out, I think, in 2013 or 2014 by CODD, C-O-D-D. Maybe it was 2013. And they found, I think it was 7 SNPs that were associated with telomere length. But combined, they explained about 1% of the variation, of course. I mean, that's just, yeah. So it didn't explain a lot, but there has been some work on genetics. There's been twin studies to do those estimates of heritability, although those vary a lot across studies. Yeah.
So, you mentioned the study about telomere length and depression,
yes.
anxiety. Are there also any other studies about the relations between telomere length and personality traits?
Yes. So there's been, I wish I could remember who they were right now. I just lost the name. But there was a study in 2006, I think that was like the first study that looked at depression and telomeres and did show that there was shorter telomeres in people who had a history of major depression. Most of these studies have been done with clinical samples, which, you know, has some issues. But Owen Wolkowitz at UCSF has done a lot of work on depression and telomeres, and I contributed to a meta-analysis that's supposed to come out pretty soon. I contributed the haines data to that. So I think there's pretty strong evidence at this point that depression and anxiety, at least like clinical depression and clinical anxiety, are related to shorter telomeres. Why is not entirely clear. Yeah. Yeah.
What about things that might buffer the effects of low SES?
Yeah, so I have not looked at that. That's a great question. Things that would buffer the impact of low SES. So I'm thinking about that for the Ad Health study. One of the things that we were thinking about was social support and social integration, since we know that there's a lot of evidence that social support and social integration buffer the negative impact of stress exposure on health outcomes. So that's a hypothesis. Coulter has that paper looking at the genetic variance and susceptibility to psychosocial stress exposure on telomere length. So there's the possibility of genetic factors that could buffer it. I'd love to hear what you think might be some potential buffers.
Coping in relationships.
Yeah. I think that's where I would start, because I think that's where we have the biggest base of evidence for other things. I mean, that's one of the reasons why when I started looking at telomere length, I said, let's start with SES if you're going to look at psychosocial exposures, because if you don't see anything with SES, you should probably move on because it's such a, robust finding in other health literature. So I think I would start with this social integration, social support types of measures, but it would be great to look at other things. Vanessa. Yeah, Grace.
So you touched on this a little bit, but when you're looking at leukocytes, I mean, you have to deal with the exposure to infectious agents.
Yeah.
And the effects on telomeres from that. Are there studies in the pipeline that are comparing different cell types to what you're finding in leukocytes to see? Because it's conceivable that some of what you're picking up on is variability in exposure to infection.
Definitely. So Allison Aiello has a paper that's under review right now with MESA looking at infection and telomere length in the MESA study. I can't remember exactly what the results are, but she has done that. And there's some interest in doing that in haines. You could do that.
Yeah, that's very interesting.
The data are available. So, I have a friend who's done some work on a study in Costa Rica, David Reckoff, who came and gave a talk not that long ago. And he has found seasonal variation in telomere length, which he thinks is explained by infectious exposures. Yeah.
Great. Well, thank you very much.
Thank you.